We are ready to begin our second session. To chair the second session, I would like to invite Dr. Gianluca Bona. He studied physics at ETH Zurich, where he received a PhD degree for his investigation on surface magnetic structures. Subsequently, he joined IBM Zurich Research Lab and did research in intense high-speed semiconductor lasers. In 1994, he did work on integrated optical devices, which led to a series of reconfigurable planar light wave circuits and photon band wave concepts. Since 2004, he is Department Group Manager of Science and Technology at IBM's Almaden Research Center in San Jose, California. I would like to welcome Dr. Gianluca Bona.
Thank you, Dharmendra. It's a great pleasure and a great honor to be here because, as you heard, I'm a physicist that still believes in the good tools that physics and mathematics have developed over the centuries. But I'm also very aware that our tools are limited and we have to reach beyond that and integrate much more from that small world of physics. And that's why I was so excited when we got the proposal to have these discussions and workshop in 2 days about cognitive computing. And I'm really pleased and honored to introduce now a person that has worked at the interface between physics, used physical tools and computational tools to bring the field of neuroscience further. It's Professor Dr. Henrik Makram. He's co-director of the Brain Mind Institute. He's director of the Blue Brain project at EPFL, École Polytechnique Fédérale de Lausanne. He is director of the Center of Neuroscience Technology. Henry Makram has been, before joining EPFL, in various research roles in the Weizmann Institute. He was an early tenure Stanley Helen Diller Professor of Neuroscience. He was also working for a couple of years as a Minerva Fellow in the laboratory of Nobelist Bert Sackmann at the Max Planck Institute. Among his many contributions to the fields, I just want to mention one. It's the precise millisecond relative timing of single pre- and post-synapses action potential to reveal a highly precise learning mechanism operating between neurons. This is what people in the field call today as STDP, spike timing dependent synaptic plasticity. So these many fields that Henry Makram have worked in has also led to a series of prestigious awards such as the Ebner Science Award, the James Heinemann Prize, the Abraham Research Award, and of course he's now leading the Blue Brain project that is connecting EPFL and IBM in this field of neuroscience, and it's for me a great pleasure to introduce Henry Makram, who will talk to us about the emergence of intelligence in the neurocortical microcircuit. Please welcome Henry.
Thank you very much for the introduction, and thank you very much, Damodar, for this honor of this invitation. So, about 3 weeks ago, we actually had a conference in Barcelona, which was to commemorate the 100 years since Ramon y Cajal started drawing every neuron in the brain. And he essentially did. He drew every neuron, every type of neuron that we basically know now. The purpose of this conference was actually to reflect back on the past 100 years, but also to look forward into the next 100 years. If 100 years can happen, well, then you can look a little bit further and try and think what's going to happen in the next 100 years. Well, two very interesting issues or topics of debate arose in this meeting. And the first one was, many people said, what would we do as mankind to put inside a time capsule and send it out to space so that it would land on some alien planet and they would be able to understand who we are? So, of course, you had all kinds of opinions. You had people saying, let's send the genome. We send it all there. So you're going to read the genome. I don't know. We don't know how to read the genome. We don't know how to reconstruct who we are from the genome. Of course, it's a very important milestone, and it's an important thing to send. But are they going to understand who we are? Other people said, because it was, of course, a meeting about the cerebral cortex, about the brain, all the different types of neurons, they said, let's send all the piles of books that show the different neurons of the brain. Okay, you could send that. We can't even look at all these thousands of papers and reconstruct the brain and work out how it works. So do we think that they're going to be able to do it? Maybe if they're very advanced and they've already seen that kind of biological design, they may say, yeah, okay, this is easy. But maybe it's a silicon race, and they can't work out how we're built. So, of course, there were also other people, and they said, let's send DARPA's robots. So if you send DARPA's robot, are you going to put your name on there and say, this is us? So, yes, they would recognize that we're a very advanced race. We can build very sophisticated robots. We could say Darwin X, and they would know that we can build very sophisticated robots. But is that really going to tell them about us? The bottom line is that what we think is that the one thing that would really demonstrate to an outsider that we really understand what the brain is doing is if we sent them the blueprints to build us. I don't mean the biological blueprints, but that we've actually understood all the biological processes to build a robot that will look like us, interact, and have all the cognitive capabilities. Of course, that's a dream, it's a challenge, and that requires to build a robot with a brain that is virtually a copy of the way that we are. And so the first part of my talk is going to try to illustrate to you the very first small step towards not just building a neural circuit that is very clever, but to building an accurate copy of a small part of the brain. The second issue, which was a very interesting issue that was raised, was what makes humans special? So you had all kinds of reactions. Some people said, humans are not special. They're not special because in each environment of a different animal, we all adapt as well as we can within that environment. But the overriding opinion was that we are special. We're special because we can share knowledge for the purpose of understanding our own brain. And it's difficult to find that animals could do that. But I think that there's another reason why we're far more special than that. And that is that imagination is imagination. Imagination is more important than knowledge, as Einstein said. And I think the reason why imagination is so important is because it allows us to look inside the paradigm within which we are investigating ourselves. And not only to look inside that paradigm and say, this is the foundations, the axioms that we stand on, that we use to interpret the brain. But it allows us to step outside of that paradigm and saying, what if we actually interpreted the brain from a completely different place? So in the second part of my lecture, I'm going to try to take you and illustrate briefly the current paradigm that rules, the paradigm from which we interpret how we understand the brain. And then take you to the opposite paradigm. So let's begin with the first, as we heard beautifully from Jerry Edelman, that the human brain really is because of the neocortex. The human capability is because of the neocortex. That is the emergence of man from mouse to man, and it's a 1,000-fold expansion in the neocortex.